स्क्रीन चेंज कर दो फटाफट. Okay, so we'll start then. I will introduce you first, and then so you take over.
Yeah, fine.
Okay. So a very good afternoon to everyone, and welcome to Vertical 2 of today's session at DDH Training 2020. Today we have with us Dr. Prakash Rathi, Senior Data and Analytics Engineer, Augmented Drug Design Platform, AstraZeneca. Prakash finished his PhD in Computational Biotechnology from University of Dusseldorf in 2014. He then worked at Astex Pharmaceuticals, Cambridge, UK as a postdoc and scientific software developer. Since December 2019, Prakash is technical lead for AI ML workstream with the Augmented Drug Design Platform at AstraZeneca. He has over 10 years of experience of developing scientific software and machine learning models for drug design. I'd like to thank Prakash for accepting our invitation for taking this session. And in today's session, he'll be talking about augmented drug discovery with data and machine learning. So I now hand over the session to Mr. Prakash.
Thank you, Hriday, for introducing, and thank you everyone in the organizing committee for giving me this opportunity to speak to you all and present some of my work, which I've been doing in the last 5 years, mostly with Astex Pharmaceuticals. Yeah, so this is a very simplified view of a whole drug discovery process on the slide. So it is very, although the drug discovery process is very complicated, multidisciplinary, very expensive, time-consuming, tedious process. But in simple terms, what you do is basically you find out a target which your disease is associated with, and then you validate this target, and you find some tool compounds that can interact with the target and probably modulate the target and change the disease condition. And then you use some biophysics to actually find some of the compounds which are hits or leads against your target of interest. You can use X-ray crystallography, more recently cryo-EM or NMR, to actually find the bound structure of your drug and protein. This gives you a clue how your target molecule will bind to your target protein and modulate the drug, modulate the disease. This also gives you an idea how you can change your small molecule such that it becomes a better binder to your target and at the same time becomes less and less toxic. So the drug discovery program starts with lots of molecule with trial and error. You try against your target before the clinic in test tube or in animals, and you find out some good candidates that you take to the phase 1. And then you, in a small set of people, you try the drug out and see if it is safe or not. And in phase 2, you check efficacy and so on. We all know it. But the important point to note is that this whole process, most of the times, take about 10 to 12 years to finish. And also, it costs about $2 billion for one drug to reach the market. The reason being, many of the molecules that enter into the clinical trial fail at different stages. And the computational tools actually try to help design better molecules which have less chances to fail in clinical trials than you would have if you had done like a high throughput screening randomly against a huge library. So I will actually focus on computational tools in this area, like the biophysics. So basically, the computational analogs of, let's say, X-ray crystallography to predict the binding pose, how a drug molecule would bind to a protein, and also how do you score which molecule out of millions that you can generate virtually bind well to your protein. So it's quite common to use some scoring functions. So here's your protein, protein which has the active site, and this is the surface on the protein, and there's a molecule, small molecule, which could be a potential starting drug or starting lead for a drug. So it binds to the active site in this way. And this is actually an image generated by X-ray crystallography. So you freeze the protein in a crystal, and then you soak it with a sample of a drug molecule, and that it goes and finds the active site and sits there. And then you take X-ray shots, and then you solve the structure. That's how this image is generated. But this image doesn't tell you much about if you had like 10 or 20 or more such small molecules. You don't know which one would bind better among these 20 or hundreds of thousand. So we use some scoring functions to actually gauge what's the strength of binding of a ligand molecule to a protein. And there are generally three classes of scoring functions. The first one is physics-based scoring functions, where you actually measure the interactions like non-bonding interactions, for example, van der Waals interactions, electrostatic interactions between protein and ligand, but also you can have bonded interactions, so bonding angle and dihedral and so on. You sum them all up, and then you get a score, and that's your physics-based force field method. There are some examples: Ember, Charm, OPLS, and there are many others. The other category is empirical scoring functions that are derived by fitting the experimental observations. So experimentally you can measure the binding affinity of your drug or a small molecule to a protein, and then you have a huge set of that. Then you can actually infer based on the observations that it makes a hydrogen bond or a hydrophobic interactions. The carbon is near this region, or there's a pi stacking, and then you sum these interactions, and then you try to fit the experimental observations on a setup molecule, and then you generate some empirical scoring functions and use to actually predict binding affinity for new drugs. The last class among these three is statistical, or otherwise also called knowledge-based scoring function. Why they are called knowledge-based is that you have a huge knowledge of protein ligand complexes. As I said, this particular picture is generated using X-ray crystallography. There are many such protein ligand complexes have been solved and deposited in PDB data bank. You can take them all, and then you can observe that there is some pattern which emerges, that some atoms want to come near to some other specific atoms. For example, partially negatively charged atoms like to be close to partially positively charged atom because they make hydrogen bond or better electrons interaction. Similarly, the lipophilic carbons would like to be close to the tryptophan residue in the protein because that's more lipophilic. So these kind of atom-atom pair potentials you can generate from this knowledge that we have generated over last 50 years or so. So I will talk about one of such statistical potential or knowledge-based scoring function called PLIT that we developed at STEX. So if you want more detail, of course, you can go to this paper and see, but I'll try to cover some of the basics of PLIT scoring function here. So in a one-slide view of PLIT scoring function, how it works is that we take all the PDB entries with protein ligand complexes. So here's one example where there's a protein, and this colorful sphere style displayed in the colorful sphere style is the ligand. So you have one example here, but you have about 80,000 plus such complexes in the PDB. You take that, and then what you do is basically for each atom in the protein and ligand, you type them, you classify them. You say, here's the carbon, here's the oxygen attached to nitrogen, or here's the amide oxygen. You can classify them in different groups based on their connectivity. Once you have that, you actually try to calculate contacts between protein and ligand. And here's a nice way to actually quantify the contacts, or actually segregate the contact of a ligand molecule and partition them into pair contacts. So basically, we can answer questions like how much this atom is contacting to another atom of the protein. So basically, you can do a Voronoi tessellation, which is basically partitioning points in 3D space. And once you do that, you can actually see the pairs directly here. And for each pair, you can calculate the contact area. So then you can tell for my ligand, this atom is contacting this much to this protein, and rest is contacting to the water, and so on. So you have lots of observations for different atom types. Then you can actually generate two types of propensities. Propensities here means actually probability of being close together. So here, one is the atom type propensities, basically telling the carbonyl oxygen atom type how likely it is to be near amide nitrogen. And it turns out it's quite likely because they form hydrogen bond. And similarly, you can have a geometry propensities, which tells you what are the distances, what are the angles they like to interact. When they interact, what are the angles and distances they would like to form most often. And here's some more detailed view. So here's our ligand in the center, and these are the protein atoms. You do a Voronoi tessellation to partition them, and then you calculate the areas marked here. For example, this is the intramolecular contact. This one is with the carboxylic oxygen, and here's the pyridine nitrogen interacting with that. So basically, you can see this area basically tells what is their interaction. These two atoms, what's the area of interaction there, and the area which is not actually interacting with any other ligand or protein is actually solvent exposed. So you can find solvent exposed area, and then you can also set up a local coordinate system which will guide you. Distance is quite simple. Of course, you can draw a straight line between ligand atom and protein atom, measure the distance, you know how far they are in the complex. But for angle, use polar coordinates. We define two angles, which will tell you the specific direction in which this atom is based on the local coordinates. And when you have data about all the complexes in the protein data bank, you can generate the propensity map. So here it is showing you how likely is the oxygen, sp2 oxygen, interacting to another sp2 oxygen. It turns out they are not, because they don't like each other. Whereas the oxygen would like to be near, probably near nitrogen, let's say pyridine nitrogen or pyrrole nitrogen, because they form good hydrogen bond. So here, and similarly, you see the lipophilic carbons, they tend to be close to lipophilic carbon, and that's why you see a better interaction or more frequent interaction of that type in the PDB. Similarly, you can see distances. So some contacts like to make a short distance contacts, for example, hydrogen bonds with amide hydrogen and carboxylic oxygen. You see that they form their most frequently observed distance is around 2.5 to 3 angstrom, whereas some non-specific interactions like methyl to methyl is actually they happen at the longer distance. Similarly, if you see here these angle preferences, again the hydrogen bonds they make they are quite particular about what angle they make interactions with. Mostly they are straight to the face direct interactions with 0 degree in our local coordinate system. Whereas if you see CH3 to CH3 interaction, they are non-specific. They actually come around from all the side. It doesn't matter. Whereas hydrogen bond, they straight come down. The hydrogen would directly point to the oxygen of the other atom. So this is the information you get. What you can do is actually sum this information and form a scoring function telling, if you invert this, you say, okay, if this is more likely, then if you see this information, this type of interaction in your protein ligand complex, it is a better, in energy terms, it is a better interaction. And then you can use that information, use that scoring function to actually test how it performs. So this scoring function, we compared against lots of other scoring functions that were out in the scientific domain. So here I'm showing the top. I'm showing the performance in the docking. So to find at the top ranking pose predicted by our scoring function, what's the fraction of complexes that we predicted correct. And the PLIT perform as well as other well-known scoring functions on many docking exercises. but it performs exceedingly well on fragment set and drug-like set compared to the other docking scoring functions. Similarly, we performed this analysis on a virtual screening. So virtual screening is an experiment where you have thousands or millions of molecules, virtual molecules, and you run your scoring function against each of those molecules and tell what the docking score for each of the molecule. And then you rank them from the best scoring one to the last scoring one. And then you actually can create such a plot which will tell you whether your scoring function is as good as random, which will be like this line. But if it is above the random, it means that your scoring function is ranking correct complexes at high rank. The complexes that occur in the nature or which bind at high energy actually are ranked at high with higher score. So the further up you go from this diagonal line, better you are. And PLIT here again does as good as other scoring functions for the dead set. But again, in the fragment set and drug-like set, this does really outperform other scoring functions. So this is about PLIF, but I thought that I will show you a quick demo how you can actually use it, because we have made it available as an open source. I will show you a quick demo. You can just grab the code from here and run some examples and see. So first I will show you the protein ligand complex A. So here's an example protein, and let's see that as a cartoon, right? And then here is an example ligand. So you can see the ligand binds. Again, this is a known protein ligand complex in the PDB, and we are just scoring this. And this is a cartoon representation of the protein, and here's your ligand, how it binds. But as I said, this complex doesn't tell you much about how tightly it binds and so on. And using our PLIF scoring function, you can actually score them. So we have this. Repository for PLIF on Bitbucket that you can download and actually use. So take this URL and then you clone this repository. Run the command to build the program. And then you just set what is the PLI directory that your program is in. Then there is some example code here that you can run. For example, you can run according this to complexes. There's an example file there. You run it and you get a JSON object which tells you the PLIF score for your complex. And if you had many such complexes, you can run all of them, and then you can compare how good or bad they are, and you can rank them. So this was about PLIF. Let's see some other uses of PLIF. So we actually used PLIF to predict fragment binding. So basically, in drug discovery, fragment-based approach is the one where you start with very small molecules rather than drug-sized molecule to screen against your protein. And once you find a very small ligand hits, you actually try to optimize them by adding small functional groups to them. And we actually developed a PLI map, which actually predicts where on your protein these small fragment-sized molecules would like to bind. Again, you can see the details in this paper. I'll again cover the basics here and show you a demo. So what is actually fragment mapping? So you can do it experimentally using X-ray crystallography. The idea is that you freeze your protein as a crystal, and then you keep on adding a high concentration solution of many small molecules. And then you solve each of those structures and you see, oh, there are benzene molecules binding here, acetone binding here, and so on. So you can actually see, you can map whole of the protein where things bind. And the places where a lot of things bind are called hotspots, because for the obvious reason that many molecules bind there, and they tend to be kind of a good starting point from where you build your drug molecule and grow this small fragment. And the PLI map is a computational analog of this. How PLI map works is that you take your protein structure again computationally, and then you place a regular grid over your active site, and then you take your fragment molecule, benzene here, and then you place it on each of the grid point in a sequence, and also you sample the orientation of this molecule on each grid points. So basically, walk on the grid points, keep rotating, and each time you do, actually you calculate this score. So the PLIT score, which I showed you a couple of minutes ago, is performed at each combination of this virtual pose that we generate. Once you have millions of such calculations done, you can actually find the clusters where this benzene molecule has very high score against this protein. And then you actually can see, oh, this is the best ranking pose, and there are many molecules binding there. And then you can run it against many different fragments, and then you can get a very fair idea about which is the hot site on your protein and which is the warm site, and so on. So we actually compared it against the experimental findings. So one of the examples in PKB protein, the Abbott molecule binds nicely, and at Aztecs we performed experimental high concentration search, and we found that pyrazole molecule actually nicely maps this molecule, drug molecule, or drug-like molecule. This is actually experimental finding, 3 molecules, and we run our program, and they actually nicely map these 2 sites, these 2 pyrazole sites using this computational tool. Although this third site is not mapped, however, this third site is actually formed by these 2 other sites, and we run these simulations 1 molecule at a time. We don't know the availability of other 2 molecules. Similarly, on another example from MDM2 P53 protein, which is a target for cancer, our tool nicely maps the hotspots on the protein. We also actually evaluated our fragment mapping tool against a high-quality fragment mapping set, although such a set did not exist, so we created one again using the PDB. So in summary, actually, we took the PDB, we chopped off all the molecules, small molecules in the PDB virtually to form small fragment molecules, and then for a given protein molecule, we aligned them all together, because the same protein can have many structures with different ligands. What you will see is that, oh, here the sulfonamide, benzene sulfonamide group is quite prominently sitting here, and it is being placed here by many different molecules. So this would be kind of a hotspot for benzene sulfonamide. Similarly, you can see chlorophenol sitting in another area of the protein. This can be another hotspot or warm spot. So we actually constructed 52 unique protein fragment complex set from the PDB, and then evaluated our program against many different scoring functions that are out there in the market and many other techniques. So here is the baseline technique. It's called LigSite. It's very simple, not very, it doesn't have sophisticated scoring functions, and doesn't take into account the interactions of the protein and ligand. It doesn't do well, but not surprising. It is a cavity detection program. It finds it, but it doesn't find all the time. Then there's other program that we developed which finds 40% of the time correct, and then there are some state-of-the-art scoring function that perform above 60%, and PLIT actually does as well as them. So PLIT being used in a goal docking does as well as, but our own tool with PLIT scoring function, PLI map, also does well. So, however, how often do we get right is one thing, but how correct you are when you predict a docking pose. If I go back here, we actually predict these poses using PLI map, and if they are very close to the actual pose in the PDB, we say your answer is even better, not just within one and a half angstrom, but if we go closer to the binding pose in the PDB, then the quality of your prediction is even better. So we actually outperform all the scoring functions, the quality of the score. So our average error in the prediction, or average deviation in the prediction, is just about 0.4 angstrom, whereas others is around 0.6 angstrom. And this is quite important in drug design to get a better binding orientation, binding pose, so that you can design new molecules. The other advantage of our PLI map offers is that since we actually scan whole of the active site, not just randomly choosing the spots, we systematically scan whole of the active site. We know at each point what is the score for your probe molecule. So you can actually generate nice maps telling, oh, here are the areas which are most likely. So this is the probably best area to be, then this is the next one. So it also finds the other areas where there is a likelihood that this group will sit nicely. So not only does it find the hotspot, it shows you, it gives you the whole picture of the active site. The warm spots and hotspots, what are those actually? As I said before, if there are many ligands placing the same thing in the same place all the time, then it is quite hot because it's basically all the ligands place this group here. But sometimes you see a warm spots where not all the ligands, but yeah, about 50% or less, if they place, for example, chlorophenyl group here in the glycine rich loop of PKB, which means this is kind of a warm spot. So we categorize them if, let's say, 80%, more than 80% of the molecule place a group at the same site, then it's hot. And if it is less than 50, it's greater than 50, but less than 0.8, then it's warm. And in our example, it is quite intriguing to note that our program actually works very well against the hot spot, but against the warm spot, it's slightly poorer than the other scoring functions. And it is true with the top 3 ranks as well as top 1 rank. And the reason is that since we actually search the whole of the active site, we are quite good at finding the best ranking spot because we don't leave any stone unturned in the active site. There are some challenges in the validation side. These are common challenges across all the scoring functions. This validation set is quite tough nut to crack. For example, we use RMSD as a measure of success, whereas this has a RMSE greater than 1.5 angstrom because this chlorophenyl group is actually flipped. Whereas this makes a hydrophobic interaction, which is non-specific. So this is not a bad prediction in terms of the location it has found. But if you use RMSD as a success, this will be a failure. Similarly, some water-mediated interactions are not captured because we don't have information about water. So without the water, we get it right, it is flipped. But if there's a water, then there's more likely sit in isoxazole, so more likely sit in this orientation rather than the other one. Similarly, protein, there are conformational changes. So we use one, so in case of HSP90, we say this is the hotspot, this actual hotspot is here. However, this protein, this cavity is not formed in this structure. It only forms when the ligand goes there. But we have a static structure to start with, and this is quite a challenging task for all computational tools. And sometimes there are some nearby hotspots. So this is a warm spot, and we try to predict chlorophenol. Where does it bind to sit? So it finds a legitimate site here for chlorophenol, because you see in the lead molecule there is a phenyl group here, and chlorophenol should also go there. But if it is already occupied, then chlorophenol would also go there. So that sort of challenging cases are there in this set. So I'll give you a quick demo using the same software, basically. So you can run this using the same PLI tools that we have built. The fragment mapping example. So this is a simple. Oh, sorry. Yeah. So this runs here, and this generates some poses that you can again see in the visualizer that I showed you before. So there's our protein molecule again, and maybe again see cartoon. And that's where it shows where your benzene molecule would like to bind. So you see all the clusters shown here. Okay, let's move on to the next item, slightly more exciting because this is hot these days, deep neural networks. So what I will show you is another way, another tool that can be used in drug discovery, which is a drug design tool, which is electrostatic potential surfaces. And we developed a deep neural network to actually predict those surfaces quite quickly. So before I go into the actual work, I thought that it would be a good idea to give an introduction to deep learning, what it means. So actually, AI or deep learning is everywhere these days. You say, OK, Google or Siri, or naming children's, or synthesizing music, or natural language processing, diagnosing your X-rays, and so on, are actually generating arts. So this image actually is generated by a deep neural network and has been sold for $432. So AI is everywhere. However, there's a hype cycle where each new technologies go. AI is still not at the AI, I mean, general intelligence is up there, but the deep neural network is at the peak of its hype cycle. The hype cycle by Gartner actually tells the life cycle of each new technology. So what you have is basically a trigger where you start a new technology, and then you become too much excited, and then there's a huge hype around that. That's called the peak of inflated expectations. But then soon you realize that, oh, it's not going to solve all the problems in the world. And then you reach disillusionment when you say, oh, this is not useful, it's overpromised everything. But then as science progresses slowly, people will find the useful examples where this technology can be applied, and then you have a plateau of productivity. Then you have a slope of entitlement. You find the use cases of all the technologies. So the good thing is that it's at the peak of hype, but then it will sooner come to each one has its own pace when it comes to, and AI and deep learning in drug discovery has already shown some promises. There are a couple of papers out there that have created drug-like molecules out of AI. So we are already, this is actually 2 years ago, we are already somewhere around here. And there are many pharmaceutical companies that work in the area of the AI. I'm not even talking about the proper pharmaceutical companies. These are AI-based pharmaceutical companies that have AI as the main focus. They work in the areas of drug design, chemistry, bioinformatics, clinical area, or data analytics, and many other areas. And in drug design, if you see, there are many different applications. You will see papers coming out every day in different areas, ranging from affinity prediction to toxicity prediction to generating new molecules, planning synthesis, and so on. And similarly, you see a quick jump in papers that mention machine learning. It's also normalized numbers, like papers per 100,000 is consistently increasing since 2000 or so. And there's a resurgent machine learning coming up because of the availability of fast GPUs. So I thought that it will be good to actually demystify some of the buzzwords, because we hear AI, we hear deep neural nets, we hear many other terms, machine learning, what they actually mean. So there's actually, if you call this deep neural network, which I'm going to show you, if you call that AI, you're not wrong, but you're not specific. AI is a general term, which can have expert systems, knowledge graph, or machine learning techniques like random forest or supervised, unsupervised. This is like an umbrella term for technique by which machines learn to do tasks that are otherwise done by humans. And then machine learning among the AI is basically you have supervised learning, where you have some examples. For example, you have a set of patients for which you have quite amount of data, big amount of data, and then these are patients, and then your other set who are not affected by the disease, and then you can build a model which is a supervised model. You feed this training set that here are the patients, and here are their descriptions, here are the non-patients, and here are their descriptions. And the AI is a subsection of machine learning. Artificial neural network is a subsection of this machine learning, where you use such an architecture where you feed in data, and then you do some mathematical calculations, and then you get some output. So this feed-forward network is one of the form of the AI, and deep learning is the one where you have many, many layers of such mathematical calculations going from your input layer, which is your features, basically telling all the patient data, and here's the output, which is what you're trying to predict. So, and you have many different layers in between. Then it is called deep learning. And then there are some specific variations of this deep learning, which we are not going to talk today. And so how do deep neural networks work? Basically, there's a huge amount of data. You feed the data at one end and keep digging around. And then at the other end, you get the answers. That's a funny description of the AI or machine learning in general, but not far off from how it does. So here's one toy example. So you have 5 persons, and some of them are diabetic, some of them are not. And they have some descriptors. Descriptors here are the factors that probably determine if you will be diabetic or not. Let's say obesity, exercise, and smoking. 1 and 0 here means this person is obese and doesn't do exercise, doesn't smoke, and this person does exercise, and so on. And here's the outcome, diabetic. If we know this is our training set, how deep neural network works is that it actually, you can solve this by multiple linear regression by adding weights to each of those factors, and then you can predict the diabetic likelihood. But the deep neural network actually does it by this architecture, where you feed in your input and you try to predict the output. And each input, you basically sum these factors into the next layer by adding some weights to each of those. And then you sum, and whatever the value is, you get the output from there. And this is, let's say, step 1. You try a random weights, 0, 0, 1 on these things. and you try to predict. This is the actual state of the first example. And with this set of weights, you say this is not diabetic, but actually it's the wrong prediction. Similarly, if you keep on trying different examples in your set with this set of weight, you get some right, some wrong. So computer tries another step. Okay, let me change the weights. It changes the weight. It gets some more right, some more. Tries another one, gets some other right, another wrong. It keeps on trying until it gets the answer right. And probably at some step, it will find a perfect set of weights which will describe the diabetes status for the set as accurate as possible. However, this is an oversimplified example. The deep neural network does it changes these weights intelligently by going in a direction which will make it faster rather than randomly changing that. And also, rather than simply summing, you have some nonlinear functions in real deep neural networks rather than simply summing them off. So this is like 101 for deep neural networks. And then I will go back to the work that we have done. So going back to my work, so in the drug discovery, ESP surfaces, electrostatic potential surfaces. Quickly, let me tell what they mean. So here is a small molecule, which is a starting hit for a target, XIAP. And this is not quite potent. It can't be a drug, because if it were a drug, you will have to feed like 10-50 grams of this molecule to actually attain the concentration so that it can bind to a protein. And at that concentration, of course, it will be very toxic because it will bind to a lot of things. So you want to improve its binding affinity against the XIAP. So one way is that you actually can see, if you calculate the electrostatic potential, which actually tells you where the electrons are in your molecules, because each atom has a different electrostatic different affinity to the electron. They can draw electron towards them. Some areas will be electropositive, other areas will be electronegative. So here, if you see, there is the electronegative area of the ligand. This hit is actually quite close to the electronegative area of the protein, which is not a good thing. You like to have electropositive near to the electronegative and vice versa. So this actually, if you calculate these surfaces, you get this insight. Oh, we need to change something here in this molecule to make it more potent. And you do it. You add a chloro group and add a nitrogen, insert a nitrogen in the ring, make this new molecule. You withdraw electrons from the ring, it becomes less electronegative, and then this clash of electronegative is actually removed a little bit, and then you have better potency here. So higher this number, better it is. So this actually is a better starting point than this. So this kind of insight comes from this electrostatic surfaces and looking them into the molecular viewer that I had shown you before. However, how do you calculate such things? So you can run a complicated and time-consuming quantum chemical calculation on this molecule and some slow calculation on the protein molecule to actually generate those surfaces. For this molecule, on fast computers can take several minutes at a very accurate level of theory to generate what was actually originally generated. That's the problem. So if you want to try 100,000 molecules, you can't run every molecule for a few hours and get these images back, because that will take lots of time or a huge amount of computing power on your data center or computing center. So what you can do is basically, you can learn a deep learning model which can actually generate these surfaces for you in a fraction of a second. How do you do it? You basically derive a model. You create a big training set of molecules for which you actually calculate these surfaces. So in our example, we had 100,000 different molecules for which we actually generated these surfaces using very slow and accurate quantum chemical calculations. So that's your training set. And then you derive a model, and then you actually test it, how it performs. So this model then can predict your surfaces for new molecules, which were not part of the training set. And then you compare how good they are from if you compare it against the surfaces that derive from a slow calculation. So we had a model to fit these surfaces. So the model was quite simple, that we place partial charges on the atoms and also non-atomic centers, for example, on the lone pairs or p orbitals or hydrogens. So we place partial charges, and then we solve this Coulomb equation, which will fit your surface nicely. So this is actually generated from the QM, and this is generated by fitting partial charges on this. So basically, for one number which tells how much charge you have, and then you fit those numbers in here, and then you calculate the surface here, and then you can compare, and they look quite the same, I would say. There's some differences which you can calculate. If you take a difference of these two, you get root mean square error in the prediction, and then you can see how good your model is. Although we had employed a slightly complicated charge model, not just a simple charge model, but a constrained charge model, which I will not go into detail today. But so what the performance is, this is basically the baseline. So here, the lower you are, this is basically an error, root mean square error, and this is the fraction of molecules in your test set, how good you are. So left side of the plot, you're good, because most of the molecules have very small error. And this is not our model. This is simply the fitting of the charges and comparing against the QM calculation. And the fitting is quite well, around 2 kcal per mole. Error is quite well tolerated. And how would our model work? We will see. So we had like 100,000 molecules. We built a complicated graph convolutional deep neural network. I'll probably go into detail a little bit about how the graph convolutions work in the molecule space. And this model then predicts, given your molecule, this surface, right? So here's how it works. In case of images, the graph convolutional neural network works by actually summing the nearby pixels in the image, values from the nearby pixel into the central pixel. So if you do that, similarly, you can do it for molecules. You have an atom here, but they are neighboring atoms. You take the properties from these neighboring atoms and sum them up into the central atom. You do it again and again, many number of times, then you get the information, each atom gets the information from the neighboring atoms as well. So you know more about the molecule. The machine learning models knows more about the molecule. That's how you do convolutional neural network on a molecule. So this is what our predictions are. So this is basically telling, this graph is telling the correlation between the fitted charges and our predicted charges. We're not talking about surfaces as of yet, just the charges. And straight line means, yeah, they are quite well correlated. And this is a density plot, density correlation plot rather than simple correlation plot, which means that the yellow pixels here have a lots of examples underneath, about more than 3,000 underneath, whereas these points, which are slightly off, incorrectly predicted, there are only very few points like that. And then let's see how well we predict the surfaces themselves using our charges. And this was the baseline, remember, from the fitting. And this is our model. And we do quite well. And how much time does it take? In this calculation takes about 1 hour each molecule, and this takes about 10 molecules each second. So 1/10 of a second to calculate this, and it's quite close to that. Compared to other semi-empirical quantum chemical calculations, for example, AM1, they are quite far off. They are their average error is about 8 kilocalorie per mole, which is far off from our model and the baseline. And what does it mean when we say like 3 kcal per mole? So here's the prediction. If it is within 3 kcal per mole, they look quite the same. This is the QM prediction. This is the our deep neural network prediction. This is quite the same. But if you look at the higher end, when say about 6 kilocalorie per mole error, Then also qualitative, the surfaces still reproduce the charge distribution of the molecule quite nicely. And for interactive drug design, when you see the structures in there, it's quite tolerable at this level. And 99% of all the molecules in the test set, we actually get at least this performance, if not that. And we use our ESP model to predict some of the chemical properties of the molecule, like pKa or hydrogen bond basicity, for many different series of compounds. And it is quite promising to see that the predictions from our ESP DNN model are quite in line with the experimental predictions for pKa and hydrogen bond basicity. And pKa is quite a useful descriptor, useful property for a molecule to know in a drug discovery program. Similarly, if we want to understand SAR, like how drugs interact with protein, it's quite useful to see that this is the area interacts with the protein, and you can nicely see that here is the interaction that is happening to this tryptophan of the protein, and you can design molecules accordingly. So we can also predict binding affinities just simply looking at the values on here, which directly interacts with the critical part of the protein. So you can actually get these values from here and plot against the IC50 of the known sets, and this works quite well for the examples we tested. So when your binding affinity is driven by electrostatic potential, this is quite handy tool to actually use as a surrogate for binding affinity for design molecules. Rather than using scoring function, this could sometimes give you better results. So just some thoughts on why did graph convolutional neural network work better for atomic charge predictions. Some of the reasons are that the atom charge depend on the environment of atom. So, and graph convolutional neural network, as I said before, actually learns about all the atoms that are in the vicinity of the atom itself. And that's why it has better chances to actually predict the properties of an atom. Similarly, so the nearby atoms that affect the charge on the atom, they're readily automatically picked up by the graph neural network model. And thirdly, we are in a complete control of the training data here. If you had noticed this learning exercise, our training data was not generated by wet lab experiments. So actually, we are not limited. We could generate as much as data we want, because generating wet data cannot be possible to cover all the examples, whereas computationally generating these QM to run QM calculations on hundreds of thousands of molecules is possible on very high performing computers. So we were in complete control of the training set. So we covered the training set in such a way that you had examples in the training set to cover a quite diverse chemical space. And to some extent, the models actually learn physics because it learns the atomic environments. And summary of this work. So this was a very, very fast convolutional neural network. As I said, you can do 10 examples in less than a second, and then you can see the surfaces nicely. And again, this whole code is available as a GitHub repository. And I will show you a quick demo, probably for this as well. So you pull this repository, and then you can actually run this program and pass on your ligand molecules, which I'm taking example molecules here. Now it is working on each of the molecule in there and generating APQR files. And . So these are the PQR files which you can use to view your surfaces. And let me show you again an example. How would you view those surfaces? So you load the molecule with PQR. So you see here atoms, but you also see non-atomic features like p orbitals and sigma holes and lone pairs and hydrogens. So you generate a surface, add a surface representation to this, and in the surface, maybe you use the surface type AV, which is STX viewer style surfaces, and radius, take explicit, looks nice enough. And then you can say color scheme can be electrostatic, I guess. Yes, electrostatic. So now you actually see, now you can see which part of the molecule is electronegative and which part of the molecule is electropositive and so on. So you can use this tool as well, it's free. Right, so I think I'm getting quite close to the finish line, except that I have a few general observations and comment. Some insights are on the machine learning. These are quite important. Most of them are known by most of the people anyway, but I thought that it's a good idea to actually reiterate them. So there is a fundamental principle in the machine learning that you have a training set, you learn a machine learning model, then you have a model, and then you hope that this will also apply to the new examples. So if you take the simplest toy example of the diabetic patient and their exercise, obesity, and those habits, you build a model and hope that the new people new subjects in your study will also follow the same train. But it is not always, true that you will achieve this generalization. If your new examples are strikingly dissimilar to your training set, then you have a problem. Your predictions will not be right. This is a famous saying by George Box that all models are wrong, but some are useful. What does it mean is that we can always say, oh, your model is wrong because it does work 70% of the time. But the thing is it better than random prediction, or is it better than a human prediction, or can it run on million molecule in 10 minutes, which human would take 10 years to do? So it can still be useful even if the model is wrong or not 100% accurate, because no model can be 100% accurate. That's why they are called model. Right. And then there's a well-known saying in the machine learning area that garbage in, garbage out. So if you feed a garbage data, you'll get the garbage out. And this also applies to deep learning. Don't think that the deep learning is a magical tool that will actually not need high quality training data, and it will just build magically a nice model. And actually it applies more to the deep learning because they are susceptible to overfitting more than some of the other classical methods, because there are lots of parameters to train here. Richie Bellman said the curse of dimensionality. As I said, the deep learning, you have many different weights. In the toy example, we just had 3 weights, but in my ESP DNN model, I had 50,000 different weights, which we optimized as part of the graph convolutional deep neural network. So think about 50,000 different parameters. It's quite easy to actually fit into that 50,000 dimensional space, overfit to your training, and then it will not work to your training data. So it's quite important to have huge amount of training data for deep learning because there are huge number of parameters. Also, model interpretability. So simple models are better because you can interpret them. You can tell. Again, I'm going back to the example of my simple toy example of diabetes. If model was that simple, if you build that model, if you see the weights, you can tell, oh, okay, obesity is bad for diabetes. If you're obese, more likely you're diabetes. Okay. But if you have number of parameters and number of connections in the deep neural network, you can't actually tell. If it is a complex model, you can't actually tell what's going on there. You can predict the outcome, but you can't learn from that prediction. So interpretability is inversely proportional to the complexity. More complex the model, more difficult it is to interpret. Right. So just we'll show this very striking XKCD comic about the modeling, and another nice paper. So here, if you see, people say data don't lie. You have data, but actually models can lie. If eagle eyes amongst you might have noticed that all the plots have the same underlying data points, but you can fit many different things. You can have quadratic, logarithmic, or logistic, or whatever. It's quite important to not overfit your data points with a function which fits nicely to your data, but then outside of the data doesn't do anything nicely. Any new example comes, and then it fails. And as I said before, less number of parameters, better it is. More parameters you have, larger training set will you need. And this is a very famous saying, which is quite often used in the used by physicists or mathematicians to quote about the number of parameters in a model. There's only one Newman. He was like one of the most famous physicists, mathematicists, computational scientists. He once said that with four parameters, I can fit an elephant, and with 5, I can actually make him wiggle his tail. And here's a slightly, I find it funny, paper which actually shows that you can fit an elephant with 4 complex parameters. So of course you can fit any underlying training data with hundreds and thousands of parameters. The thing is, does it work outside your training set? And I'll leave this insight with you. It's again a funny, you might have noticed I'm an XKCD fan. So again, you can talk about any technology, just use this sentence, replace this word artificial intelligence, and just use this sentence, and the other person will think that you have quite insight on this technology. And for AI, we can see before we rush to adopt AI in each area of the life, we should stop and consider and blindly think about and consider before we give such a central position in our lives to the AI. So with that, I will thank quite a lot of people. The first row people, my colleagues from STEX, Marcel, Chris, Fred, Rich, Paul, and some of my colleagues, people who I work with from Cambridge University, Andreas, Richard, and most importantly, my supervisors who gave me training, Professor P.V. Bhatam from NIPER, with whom I did my master's, and Holger Golke, with whom I did my PhD in Heinrich University, Düsseldorf. And thank you everyone who has joined online, or thank you if you're watching it offline later. And thank you, organizing committee, Hridoy, and everyone else whose names I don't know, for giving me this opportunity to talk. I'm ready to take questions now. I think my slides are over.
Thank you, Dr. Prakash. It was a really wonderful talk, and along with the demo that you have made, it really informative. We do have some questions with us now, so I'll like to hand over to my friend Lizzo. He will pick some questions. So Lizzo, if you could please pick the questions.
Yeah, thank you very much, sir, for that most informative sessions. So I would quickly like to take a few of the questions that we have received on YouTube. So the first question is asked by Mr. Ashish, and he is asking, as the usage of multi-drug combinations is increasing, can the side effect of the molecules be predicted using any of the deep learning approaches?
Yeah, yes, thank you for the question. Yes, and actually very insightful question as well, because the more drugs you give at the same time, each individual will have some side effect, and then the sum total of all the side effects can be quite challenging to manage. Yes, so as I said, deep learning can be used as one of the examples I had shown before, that it can be used to do toxicity predictions. So you need to have quite a big amount of toxicity data, which you can actually get if you are in a big pharma company, and then you can build a model against, for example, a classical example is a hug, with which you get, if you interact with the hug and inhibit the hug, then you get cardiotoxicity for many molecules, and some of the drugs have been off market. So it's a classic example where we routinely apply hug prediction for our molecules that we design, and we don't carry them forward if they show very poorly in those models. So, yes, this can be used.
So the data set that we have to use for toxicity prediction is always a standard, like for example, TOX21 data set?
Yeah, that can be one avenue where you can find, but you can also look into the Campbell, because Campbell is a resource for protein ligand binding affinities. And if your target of interest is a toxicity target, for example, hug is a protein, so you will have quite some data there. Only thing is that you have to clean them up because they come from quite varying different sources. You can also look at the.
Yeah, thank you, sir. And another question is why graph convolutional neural network is preferred over convolutional neural network for prediction of molecular properties?
Okay, so it's not, these are not two different things, because on the molecules you can only do graph convolutions. So okay, let me go to that slide perhaps. Right. Okay. So the convolution you can do on molecule, because molecules are represented as a graph. Each atom is connected to other atoms, neighboring atoms. It is a graph. If you have to do convolution for an atom, you have to do graph convolution. You have to do graph convolution for a molecule to get the information from the neighboring atoms. Does that? Yeah, probably that answers the question.
Thank you, sir. And another question is, what are the different types of training data set that can be provided for learning, for any kind of machine learning?
Okay, so few of them I had mentioned. For example, if you are looking into predicting binding affinities, so there is a very well-known directory of decoys, DUD. It's DUD dataset, which will give you a dataset for virtual screening. Then there are docking datasets. For example, STEX created one, which is famously used by all docking software, like STEX diverse set with 100 complexes there to validate your set, but you can use DUD to actually train. But then you can, as I said, you can also generate the training set yourself. For example, if you want to build a 3D deep neural network with protein ligand structures in a 3D complexes, if you want to feed that information, you can generate those docking poses yourself using another software, and that will become your training set. And affinity data you can always get from Campbell and other sources.
Thank you, sir. There's another question, and it is asked by Mr. Dilip, and he is asking which machine learning algorithm was used for the prediction of ESP?
Right, it was deep neural networks. So this is graph convolutional deep neural network, and this works by you feed your input here, and then you have quite lots of weights that connect to the next layer, and then this feeds to the next layer, and then you also do graph convolution, as I said, and then in the end you get the output, which is the partial charge. So this is the algorithm, which is called graph convolutional deep neural network. Probably on the comments, YouTube comments, I'll share links to these papers if you want to go and see them afterwards.
Yeah, thank you very much, sir. And now I request Dr. Mahanta for giving the concluding remarks for this session.
Thank you, Lizzo. So as we come an end to this session, on behalf of the DDH training team chairman, Dr. G.H. Sastry, I'd like to thank Dr. Prakash for first accepting our invitation for the talk and for giving such a wonderful, informative talk for this session. I also like to thank all the viewers who are viewing this session in YouTube, Facebook, and MS Teams. So with this, we come and end to this vertical 2 of DDH training program. Thank you, sir.
Thank you very much. Bye.
Bye-bye.